Perceptual systems, central visual pathways. Kendall et al. Prink. Of neural psi. Outline subcortical pathways, retina, LGN, primary visual cortex, V1. Magno and Parvo streams. Primary visual cortex, V1. Partial integration of M and P. Gateway to ventral/dorsal. Ventral pathway, V4, IT. Object processing. Dorsal pathway, mount, IP. Motion processing. Summary. Retinal ganglion cells, RGCs, quick summary. Output of the retina, optic nerve. Retinal ganglion cells, RGCs, quick summary. Output of the retina, optic nerve. Center surround receptive fields. Kendall et al. Prink. Of neural psi. RGCs, two principal cell types. Midget RGC receptive field tiling. Parietal RGCs receptive field tiling. Send axons to LGN to form Parvo cellular, P, pathway. Send axons to LGN to form Magno cellular, M, pathway. RGCs, two principal cell types. Midget RGC receptive field tiling. Parietal RGCs receptive field tiling. Send axons to LGN to form Parvo cellular, P, pathway. Send axons to LGN to form Magno cellular, M, pathway. RGCs, two principal cell types. Midget RGC receptive field tiling. Parietal RGCs receptive field tiling. P pathway characteristics. Smaller RFs, so better spatial resolution. Selective for color, red-green opponency. M pathway characteristics. Better temporal resolution. More sensitive to low light levels. Monochromatic, black-white opponency. Lateral geniculate nucleus, LGN. Thalamus is the meeting point of sensory organs. LGN is the visual portion. Receives direct input from RGC cells. Lateral geniculate nucleus, LGN. LGN receives direct input from RGC cells. Receptive fields have very similar RFs to their RGC inputs. Circular. Center surround. Reflected in anatomy. Kendall et al. Prink. Of neural psi. Optic chiasm. RGCs in left hemifield send axons to right LGN, and vice versa. Kendall et al. Prink. Of neural psi, LGN lamina, 3 layers for each eye. Contralateral equals the eye at the opposite side of brain. Ipsilateral equals the eye at the same side of the brain. Contra and ipsi inputs are segregated by LGN layers. Parvo cellular, P channel. Magno cellular, M channel. Kendall et al. Prink. Of neural psi. LGN lamina, 4 Parvo and 2 Magno layers. Parvo cellular, P channel. Similar RFs to midget inputs. Magno cellular, M channel. Similar RFs to Parietal inputs. Inputs from RGC midget cells. Smaller RFs. Selective for color. Inputs from RGC Parietal cells. Larger RFs. Monochromatic. Respond to faster changes. Kendall et al. Prink. Of neural psi, LGN lamina, 4 Parvo and 2 Magno layers. All combinations transmitted to cortex. M contra, M ipsi. P contra, P ipsi. Kendall et al. Prink. Of neural psi. Subcortical pathways summary. Unique cell types in the retina form the first stages of 2 major parallel pathways, Magno and Parvo. M and P pathways carry unique spatio-temporal chromatic information. LGN-RFs are very similar to those in the retina, one reliable synapse from RGC. 6 layers in the LGN to maintain segregation of I input and MP input. Subcortical pathways are trying to take basic features about the visual scene and reliably transmit this information to cortex. Outline subcortical pathways: retina, LGN, primary visual cortex, V1. Magno and Parvo streams. Primary visual cortex, V1. Partial integration of M and P. Gateway to ventral/dorsal. Ventral pathway, V4, IT. Object processing. Dorsal pathway, mount, IP. Motion processing. Summary. Kendall et al. Prink. Of neural psi. Laminar architecture of cortex. The primate cortex is ~2 mm thick. 6 layers within this thickness. Laminar architecture of cortex. Inputs to layer 4. Outputs from layers 2/3. The primate cortex is ~2 mm thick. 6 layers within this thickness. Feed-forward circuit. Layer 4, then layer 2/3, then next cortical area. Laminar architecture of V1. Inputs to layer 4. Outputs from layers 2/3. V1. Kendall et al. Prink. Of neural psi. Laminar architecture of V1. Spatial temporal chromatic information of visual world is still segregated anatomically within the input layers of cortex, i.e. parallel pathways are still segregated. Kendall et al. Prink. Of neural psi. Laminar architecture of V1. Finally, this information is partially combined once it hits the output layers of V1. Spatial temporal chromatic information of visual world is still segregated anatomically within the input layers of cortex, i.e. parallel pathways are still segregated. Now back to Kendall et al. Prink. Of neural psi. V1 ocular dominance. Eye inputs are also segregated at the input layer of V1. A neuron's response preference for one eye over another is called ocular dominance. In V1, there are alternating bands of ocular dominance across the cortical surface. Map. 1 mm. LGN. V1. Kendall et al. Prink. Of neural psi. Primary visual cortex, V1, orientation tuning arises. LGN inputs are not tuned for edges. V1 neurons, one synapse from LGN, have tuning for edges of a particular orientation, orientation tuning. V1 orientation tuning. Stimulus. Response. V1 orientation tuning. Classic Hubel and Wiesel model. Orientation tuning arises from an alignment of LGN inputs. LGN. V1. Kendall et al. Prink. Of neural psi. V1 orientation maps. Similar tuning within column. Columnar organization, similar response properties at all depths of a vertical penetration. V1 orientation maps. Tuning progresses smoothly across horizontal penetration. V1 orientation maps. 1 mm. Kendall et al. Prink. Of neural psi. V1 orientation maps. Continuous maps of orientation preference, pinwheels. Consistent preference though the depth of cortex, columnar architecture. Kendall et al. Prink. Of neural psi. V1 retinotopic maps. V1. Each point of the visual field maps onto a local group of neurons in V1. Retinotopy equals remapping of retinal image onto cortical surface. Foveal region uses more of V1, greater magnification factor. Classic V1 maps, summary. Ocular dominance map. Orientation map. Retinotopic map. 1 mm. Why have maps? How are they created? Kendall et al. Prink. Of Neural Sci. Classic V1 maps, summary. Ocular dominance map. Orientation map. Retinotopic map. 1 mm. Why have maps? How are they created? Nobody knows for certain. Kendall et al. Prink. Of Neural Sci. Primary visual cortex, V1, summary. Pathways segregated in Algae are still segregated at input layers of V1. M and P separated by sublayers in IVC. I inputs segregated into columns. Tuning for edges arises at first synapse, orientation selectivity. Maps, orientation, ocular dominance, retinotopy. Preview to higher areas. V1 is the trunk of the visual cortex hierarchy. V1 output forms dorsal and ventral streams. M to dorsal, mostly. P to ventral, mostly. Outline subcortical pathways, retina, LGN, primary visual cortex, V1. Magno and Parvo streams. Primary visual cortex, V1. Partial integration of M and P. Gateway to ventral/dorsal. Ventral pathway, V4, IT. Object processing. Dorsal pathway, mount, IP. Motion processing. Summary. Kendall et al. Prink. Of Neural Sci. Ventral pathway, preview. Two principal paths. V1 V2 V4 TOIT. V1 V4 TOIT. Involved in object identification. Called the wet pathway. Late stages of inferior temporal, IT, cortex contain the celebrated faced cells. Kendall et al. Prink. Of Neural Sci. Ventral pathway, receptive field hierarchy. Subcortical pathways compute local differences. V1 identifies edges. V1 projects to V2 and V4, what might V4 neurons represent? Ventral pathway, receptive field hierarchy. Subcortical pathways compute local differences. V1 identifies edges. V1 projects to V2 and V4, what might V4 neurons represent? Curvature, contour integration, foreground/background. IT represents complex objects, such as faces. Ventral pathway. IT represents complex objects, such as faces. De Simone et al. 1984. Ventral pathway, convergence of inputs at each stage. We don't know how this selectivity is built from the circuitry, but we have very general, i.e. crude, ideas. Each region receives convergent input from the previous region, for example V1 to V4, thus progressively increasing the size and complexity of receptive fields. V4. V1. Ventral pathway, convergence of inputs at each stage. Ventral pathway, convergence of inputs at each stage. What happens when we scale or rotate the image? Ventral pathway, convergence of inputs at each stage. Simple models predict that the IT neurons will no longer respond, but instead IT neurons demonstrate scale, shift, and rotational invariance. I.e. IT responds to particular objects regardless of simple transformations. Ventral pathway, convergence of inputs at each stage. Lots of models attempting to explain object recognition in the ventral pathway, particular invariance, many useful engineering applications. None of them come close to the performance of the primate visual system. 200 ms to IT spikes. Also human response time. Ventral pathway, summary. Receives input from both M and P subcortical pathways, but mostly P inputs. Progressive increase in the complexity of spatial selectivity. Neurons are ultimately responsive to holistic objects in IT. IT neurons also exhibit well-known scale, shift, rotation invariance. How does this happen? Kendall et al. Prink. Of neural psi. Outline subcortical pathways, retina, LGN, primary visual cortex, V1. Magno and Parvo streams. Primary visual cortex, V1. Partial integration of M and P. Gateway to ventral/dorsal. Ventral pathway, V4, IT. Object processing. Dorsal pathway, mount, IP. Motion processing. Summary. I am going to go through each step of these processing streams, from the retina to the higher visual areas, and give you an overview of the anatomy of the processing stage, along with the functional properties of the cells at that stage. Kendall et al. Prink. Of neural psi. Dorsal pathway, preview. The where pathway. Main input from M pathway circuits within V1. V1 to mount to IP. Mount, aka V5, neurons are highly selective to direction of motion. IP neurons tell the motor system how to respond to the environment. Kendall et al. Prink. Of neural psi. Medial temporal area, mount, motion selectivity. Motion selectivity seen at two stages of dorsal hierarchy. 1. Neurons in layer IVB of V1. 2. Neurons in mount, IVB of V1 is major mount input. So what is mount doing? IVB of V1 mount. Both stages are direction selective. Medial temporal area, mount, computes global motion. You could easily identify the motion of this object. Bourne and Bradley, on and neuropsy. Medial temporal area, mount, computes global motion. Apparent motion. Actual motion. Now, it would appear that movement is in the direction perpendicular to the unmasked edge. This is what a V1 neuron sees. V1 neurons only detect local motion. Bourne and Bradley, on and neuropsy. Medial temporal area, mount, computes global motion. Apparent motion. Actual motion. Mount neurons have much larger receptive fields than V1, so they can integrate multiple cues in the image. Mount neurons can detect global motion of the object. Parietal cortex, a ridiculously brief summary. Mount passes its output to the intraparietal cortical areas. Each IP area is responsible for a different class of movement. In addition to unparalleled object recognition, ventral pathway, primates have great hand-eye coordination so that we can use tools, or hit a baseball. Dorsal pathway. IP uses visual input to tell the motor system how to interact with the environment. Differences between dorsal and ventral pathway, summarized by lesion examples. Patients with lesions of the parietal cortex cannot properly adjust their grip, width or angle, to grab an object. But they can describe the shape of the object in detail. Patients with lesions of the ventral path cannot describe the object but can easily adjust their grip to grab it. Summary. Kendall et al. Prink. Of neural psi. Things I ignored, to name a few. Many aspects of the circuitry. Local cortical circuits. Intercortical feedback. Contextual influences. Attention. Much we still do not know. We do not have a descriptive or mechanistic model that predicts response properties of downstream visual areas, or behavior. A descriptive model would vastly transform technology, The primate visual system is far superior to anything that engineers can build. A mechanistic model is the ultimate goal, a more difficult one, as this would allow us to address neurological disorders systematically. I'm not going to go into specifics, but it's easy really for a naive observer of the data on early visual cortex to say, we already know everything about the visual system. But if we did, this world would be a much different place. That's all.